pesar de que la invitación fue muy apresurada. Hicieron todos el esfuerzo de estar aquí, les agradezco mucho. Y el día de hoy tenemos el gusto de contar con la presencia de unos colaboradores de la Universidad de Virginia Tech en Estados Unidos, quienes nos estarán presentando un seminario como parte de un proyecto de investigación que trabajamos juntos. La ponente es la doctora Emily Mevers, quien actualmente es investigadora asociada de la Virginia Tech, y brevemente les daré una reseña de su currícula, ¿verdad? Entonces, la doctora Emily realizó estudios de química en la Universidad del Sur de Florida y posteriormente sus estudios de maestría y doctorado en la Universidad de California en San Diego, trabajando con el profesor Bill Gerbic en productos naturales marinos. Posteriormente realizó una estancia postdoctoral en la Harvard Medical School en Boston, trabajando con otro profesor muy reconocido, el doctor John Clardy, y después de estar unos años ahí, consiguió una plaza, hizo un par de estancias postdoctorales y consiguió una plaza en la Universidad de Virginia Tech. La doctora tiene numerosos estudiantes de posgrado, de doctorado, de maestría y de licenciatura, y ha ganado algunos premios, tiene numerosas publicaciones en artículos del área de productos naturales, del área de química orgánica, ¿sí? Y recientemente en la Ciudad Americana fue reconocida por uno de estos artículos como el mejor artículo del año en la revista. Sus líneas de investigación son muy variadas, pero una de ellas es el estudio de 100 pies o 1000 pies, 1000 pies, ¿verdad? 1000 pies, ¿sí? Que los 100 pies tienen menos pies, obviamente. ¿Sí? El estudio de 100 pies para el descubrimiento de nuevos productos químicos con propiedades para el dolor. Entonces, welcome Emily. Thank you for being here and happy to listen your talk.
Good morning, and thank you, Mario, for the kind introduction. My Spanish is very poor, but I could pick up words, and so I appreciate that. And thank you all for being here. It's a true honor. I'm thoroughly enjoying our collaborations that I have with faculty members here, and have enjoyed interacting with the many students that were participating yesterday and going to continue for the next 2 weeks. But I'm going to talk to you a little bit about our work. I started at Virginia Tech in 2020, so everything that we've done is in the last 6 years, and is in collaboration with Paul Merrick, who's here, who's our entomology collaborator and really the expert on the millipedes. And so I'm going to tell you a little bit about these millipedes here and a couple others, but how we use ecology to try to find new natural products. And so I don't think natural products are naturally occurring small molecules needs any introduction to this audience, but I'm always fascinated to think about how old this field is. And these compounds were isolated almost 200 years ago, or 150 years ago. And so this is an ancient field, and it builds on traditional Chinese medicine and herbal remedies. And these compounds are still used today, and most of us probably had caffeine this morning. And they root in a natural source, historically in plants and herbal medicines. But if we go more modern, what is the implication of natural products on modern medicine? The good news is many of the new drugs that are approved by the Food and Drug Administration in the US and other administrations around the world are predominantly from small molecules that are found in nature. 65% of the small molecules are from natural products or their derivatives. This is not to discount the role synthesis plays. Many of these molecules need to be modified before they can be clinically used, and so we partner with our synthetic colleagues to do that. But if you look at just synthetic libraries, only 35% of the drugs come from there. This is phenomenal, and this is because natural products are evolved in nature with enzymes, and their targets in human cells are also enzymes. If this was the whole story of natural products, every chemist that was interested in drug discovery would be a natural products chemist, I believe. But we have flaws. And our flaws is it's becoming challenging to find new molecules with new carbon backbones. This is a nice review article from Roger Lennington in PNAS, where we can find more natural products by the red line maxing out about 1,500 new compounds per year. Although this is a little old, it has leveled off. The problem with these compounds is that they're not new carbon backbones. They represent minor differences between things we've already found before. And they judge this by a Tanimoto score of 0.4 or less would indicate novel. And only about 15% of the compounds that we discover each year are truly novel compounds. And so how do we change this? I think we look at where our compounds have come from. Where are good drugs from natural products? And I think it's become obvious, if you want to find an antibiotic, you need to be looking at microbes. From the marine environment, from the terrestrial environment, it doesn't really matter. In this example here, you have a bacteria in the middle of the plate and a fungus over on the side. This is a simplistic view of what might be happening in soil. There are millions of microbes that are competing for the same nutrient source. And when they compete, they evolve chemical warfare agents. They evolve antibiotics that we can leverage for the treatment of infections in humans. We can look at the same thing for anti-cancer agents. Anti-cancer agents are almost always produced by sessile or soft-body organisms. Think about plants or a marine sponge. These are organisms that can't go away from a prey. They can't move away from a bird or from a fish coming to consume it. And so what it does is it produces or it partners with a bacteria to produce these complex compounds. And these aren't the natural products. These are the drugs that have made it to the clinic. And we think about these compounds as defense metabolites. They deter predation. And so when I was with John Clardy, we always like to think about a function-first approach to natural product discovery. It is exceedingly hard to figure out the function of the molecule if you don't have any idea going into it. And so if you can use ecology, what would the system be defending itself against? Then you can pretty easily figure out the structure of the small molecule mediating that. The biosynthesis, or how the molecule is assembled genomically, is trivial in certain organisms, but still a very big challenge in animals. And so with this in mind, my group has 3 major projects. We have the Millipede project, which I'm going to talk to you today, where we've been looking for new compounds that have neurotoxic properties. We also are really interested in microbe-host, microbe-microbe interaction, and this is really for the development of antibiotics or development of or immune modulating compounds with the host. And then we have an odd project, because I say yes to working on any project. And this project here is, can we make bacteria an electrical switch? And we're using plant quinones to do that, that can transfer electrons, and that is our signal. But I'm going to focus today's talk on the millipedes. It's because it's the reason we're down here. We got funding to come and collaborate with Mario and Wendy and their groups to collect millipedes and look at millipedes in Mexico. And I want to tell you about what we've been doing in the US. And so millipedes are ancient animals, as some of you heard from Paul yesterday. Actually 450 million years ago. They're the first animals to move to land. And they evolved chemical defenses many millions of years ago. And the chemistry that they produce, as a chemist, I think 2 orders are pretty boring. That's this group of millipedes here that produce these hydroquinones or quinone-like compounds. There's about 70 compounds known in this area, but they all mimic the same kind of backbone here. And they're clearly acting as irritants. They irritate a prey. The probably more well-known millipedes are these ones that produce hydrogen cyanide. They produce it in a stable format, and then when it gets disturbed, they use an enzyme to cleave it off to generate the hydrogen cyanide. They're phenomenal organisms. These 2 groups the millipedes are several inches long. They're about that long, or can get that long. And you'll find these organisms wandering the forest floor. The one we're most interested in is a Scepter class that contains 4 orders, and it's these organisms here. The picture doesn't do it justice. These are about 2 centimeters long at their maximum length, and you'll really never find these organisms wandering around on the forest floor. They're going to be on the undersides of logs as they dry out really easily. But what was really interesting about these organisms is it produces compounds like this, benzonamine. And as a chemist, you can start to see these have multiple stereocenters, carbon-carbon bonds. And these are called alkaloids, and a very classic example of an alkaloid is morphine. And alkaloids are very good at crossing the blood-brain barrier and interacting with the brain. When we started working on this, in the order of Platydesmata, none of the chemistry was really known, but we knew from some studies in Polyzonida that there were compounds like this, and this was actually work done by Phil Cruz, a well-known marine natural products chemist in 2000. And so this connection with millipedes and their chemical defenses is ancient. This is a diagram that Paul has made, and what it shows is that as a millipede grows, it gains a segment. And in each of these segments, there are two glands highlighted in these dotted lines here. And these glands are what holds the chemical defenses. And these glands evolved through fossilized records over 400 million years ago, and they contain an ozophore at the very end. And so when the gland is disturbed, the millipede is able to secrete the chemistry. We can actually capture this. These are two of our alkaloid-producing millipedes, and you can capture this with a really tiny capillary. And I have a video of that. I thought this was pretty easy to do, and then I tried it. My students did a wonderful job at getting this video. But you can see that when they tap this gland, they get a secretion right here. What we can also tell is that you don't get global distribution of the compound at the same time. This is a very specific interaction with one gland, one secretion. We can capture those secretions, and we can show that there only are alkaloids. There's almost nothing else in there. And then there's two more really interesting things about the millipedes that we work on that are distinct from the other millipedes. These millipedes that produce alkaloids, they're social. They form multi-generation aggregations, and they generally do it around fungus because they eat fungus. They are a liquefied diet, so they have a primitive mouth. We don't know how they form these aggregations. They are blind, and so how do they find these multi-generational aggregations? And the other one that's really cool is this is a video by Paul Merrick, where he's captured one of our special millipedes, Andronathus corticarius, encasing the eggs. And so what many of these species of Colonatha do is that they brood care. The female lays the eggs, and then the male will wrap up themselves around the eggs and hold on to the eggs until they're hatched. If you separate the male from the eggs, the male will go back to the eggs pretty quickly. And they're blind and they don't see them, so how are they doing this? This must be mediated by a pheromone. And so we started thinking, we know these compounds in their glands are defensive, but could they also be acting as pheromones? And so we've been working on that a bit. And so I first got into millipedes by looking at one particular millipede, this is Ischnocybe plicata. A chance encounter with a faculty member by the name of Tappy Jones introduced me to millipedes. Prior to that, I was a marine natural products chemist and thought I was only going to stay in the marine environment. But then I learned how cool these organisms are. Tappy Jones was at a university about 2 hours drive away. He needed to use some of my instrumentation in my lab because he was at a primarily undergraduate or a bachelor's degree university. And so he brought a vial of about 300 millipedes. So this is a 40 mil vial, so it gives you an idea of how small these millipedes are. And he said the compounds in there are too complex for him because he would usually predict a structure, synthesize it, and confirm it. He didn't use isolation or NMR. And the compounds just had too many possibilities. So he gave me this vial, and all we did was take the liquid off and throw it on HPLC or an LCMS. It needs no pre-fractionation. This is the cleanest fraction I've ever seen. For those that work with microbes, you know you have thousands of compounds in here. We have about 5 compounds in this mixture from a crude extract. We could really quickly figure out their masses on LCMS, and we were able to isolate these compounds in an overnight. And once we isolate compounds, we solve their structures with 2D NMR. And I'm not going to go into exactly how we did that, but we use a range of experiments. If you're not familiar with the H2BC, it is a phenomenal experiment. It was developed in 2014, and it's really made solving structures pretty trivial. These compounds look very similar to the compound I showed you in the beginning, buzonamine. But what's different about these compounds is that they have more oxygens. They've been oxygenated, and then they've also been ligated. So they have short-chain fatty acids added to them. These are functionalities that we had not seen in any other millipede compounds before, and it is what really challenged Tappy in solving these structures, because where are these oxidations occurring on this backbone? He had 20 or 30 possibilities if you include stereochemistry, and that was not possible to synthesize all those compounds. We did elucidate the absolute stereo configuration of these compounds. I'm not going to go into it. That was a 2-year process. It's not trivial with structures like this. But I want to talk about the ecological and the biological function of these compounds. We know they're coming out of the glands. If they're coming out of the glands, that means that they're meant to interface with other organisms. And so we do what any natural process chemists do, and we grab ants in our backyard. So these are not ecologically relevant ants, but they are ants that we could easily get. But what we did was we put our compound on this disc right here, 100 micrograms. That might seem like a really high number, except that each individual millipede has about 300 to 400 micrograms. They are chock full of these compounds, so this is about an ecologically relevant amount for a single millipede. And what you can see is the ants' behaviors. They all went as far away from those compounds as possible. And when one ant got kind of close, it was just preening its antenna. And so when I talked to ant biologists, they said that the antennas have a lot of neuroreceptors on there, and maybe we're engaging neuroreceptors. We don't have a library of insect neuroreceptors that we can test our compounds against, but we do have a library of human receptors. And this is at the University of North Carolina, Chapel Hill. They have actually over 100 neuroreceptors, and if you send your compounds to them, they test them for free. And so, yeah, it's a fantastic resource. They're a little slow, so 6 months to a year is what it takes to get data back. But I'm just going to show you what happened. What they do is they test all your compounds against all the neuroreceptors. If it has binding affinity of greater than 50% or more, they do a dose dependence assay. So the data I'm showing you are only the ones that have dose dependency assays for them. And so for ichthyocytin A, we were really surprised. Sigma was the last assay data that came back to us. So we wrote the paper saying everything's dead. And then we got the Sigma activity and realized that ichthyocytin A has really low nanomolar activity against Sigma 1. And there's about 100-fold selectivity for Sigma 1 over a similar receptor, Sigma 2. And surprisingly, of the 65 other receptors, we hit none of them with any real potency. So it's very selective for Sigma 1. The other compounds are very similar, and we get a small structure-activity relationship with just our 4 compounds. If you have 2 esters on your compound, like these 2 compounds here, it doesn't matter the position, you lose about 30-fold of your activity. You're in the 300 nanomolar for your potency. And most surprisingly, if you just simply add a ketone right here, that's the only difference between here, you're inactive. You're in the micromolar range. And so we suggested that this was possibly because if you add that ketone, you lose all ring flexibility in that ring, and that there needs to be some flexibility to fit in the active site. Sigma is a orphan receptor, so it means that we don't know the signaling cascade, and we really don't know the function of what sigma is responsible for. But it's been implicated in a lot of different diseases like Alzheimer's, dementia, and pain. There's a couple compounds in clinical trials for some of those disorders. But what we needed to know was our compound activating or inactivating the enzyme? Was it acting as an agonist or an antagonist of sigma 1? And because it's an orphan receptor with no functional assay, you normally would have to put it into mice and see what the phenotype is to understand that. We didn't have enough material to put our compounds into mice, and so we tried something else. There are crystal structures of both sigma 1 with an agonist and an antagonist, as shown here. Here's the agonist that turns on the receptor, and here's the antagonist that turns off the receptor. They bind very differently, particularly the antagonists engage this beta loop right here, whereas the agonists don't ever do that. And so this is great work by Andrew Cruz at Harvard Med, who's been able to crystallize these almost impossible enzymes to get this in sight. And I partnered with Anne Brown, a computational biochemist in our university, and her student did molecular docking, where she took all of our compounds and docked them both in the agonist and the antagonist pocket because we didn't want to bias our results. But we saw the same thing in both of them. For ichthyocytin A, our most potent compound, it has the lowest free energy of binding, meaning that it engages the receptor the most tightly. This was great to see that we actually had a favorable binding affinity there. And it also, though, what was key to note is that it engages this beta loop over here. And so this would suggest that these compounds are acting as antagonists. If we look at our structure-activity relationship, the 2 compounds that had 300 nanomolar activity, they had negative free energy as well, which is great. You need that. But it's about half of what ichthyocytin A is, and telling us that they're likely less potent. And our in vitro data confirms that. And what was even more supportive of this is that the ichthyocytin, the one that had micromolar affinity, really poor affinity, had only a positive free energy, which means that it does not bind favorably under any circumstance to that pocket, so we would not expect it to engage. And so with this data, we suggest that these compounds are likely acting as antagonists. But this is challenging we really need to get into animals. But we're excited about the potency of these compounds, and I just wanted to just put one where we're kind of going, since some of you guys are pharmacologists. We're trying to see if we can push this compound towards becoming a drug in some way for some treatment of likely pain. Our compound here, our lead compound here, has a huge liability. If we were to put this compound in the human body, that ester would be cleaved in seconds. So what we did was we semi-synthesized this compound here, the butyl ether. Not ideal, but it's the easiest synthetic route we could get to not use some material. We want to try getting it to an amine. But when we do that, we increase the stability of this molecule quite dramatically. So the half-life in microsomes is about 21 minutes. Very, very poor. It gets chewed up really quickly. But if we make a butyl ether, the compound is incredibly stable. We can also test that if it crosses the blood-brain barrier and crosses the gastrointestinal tract, and it can do that as predicted. These are small molecules that are really lipophilic. And it doesn't really engage the P450s that degrade other drugs that you really can't afford to hit if you're trying to develop this. There's a little bit of a liability here with the butyl ether, but I suspect that we didn't actually get that clean because we did these reactions on less than 1 milligram. But we do take a little bit of a hit to the sigma-1 activity. We now have a new collaborator, Thomas Keck, who turns around the assays much quicker. He's at Rowan. And so he retested sigma-ichnosybin A and the butyl ether. And we lose some activity, but we think that this is a good lead and gives us information on how we can semi-synthesize our molecule to get it to improve the properties. With that, then Tappy Jones came to me and he's like, do you realize there's a millipede that lives on your campus? And I had no idea that we had millipedes on our campus. We're actually located right about where this blue dot is in the mountains. And this is when I reached out to Paul Merrick and was like, I hear there's a millipede Andronathus corticarius. Can your group help us find this molecule or this organism? And so we next turned our attention to Andronathus. And what's great about collecting millipedes on campus is that you can do it with your students. The campus can come out and take photos of you doing it. And so this is Paige Banks. She was the first graduate student to work on this project. And actually, all of the graduate students in this photo right here just graduated in May and have now gone on to postdocs and faculty positions. But when we took this millipede, we took it and we put it into alcohol. And we were really surprised when we put that extract on our LCMS. We didn't have 1 or 2 compounds. We had over 25 compounds. This was remarkable chemical diversity that was yet to be seen in any millipede in any order. And so this was really surprising. Paige did a very good job. She developed a 2-hour gradient on our HPLC to be able to isolate many of these compounds, and she solved the structures. And I'm going to summarize that in one slide here. There were actually 2 classes of compounds in this extract. And the first one didn't really surprise us. It's this compound here, this bicyclic, 5-6-membered bicyclic. And it still had been having oxidized and has been ligated with fatty acids. All of the chemical diversity in this fraction is what lipids get added onto this molecule. And so that made the separation even worse because sometimes a 4-carbon one was here and a 5-carbon one was here, and then other times they were just inverted. But what was more surprising was this other structure that she gave to me. She was really frustrated. She handed me this structure. She's like, I don't think it's right. It doesn't make sense. It's this 6-6-5-6 fused ring system. This is incredibly more complex than anything we've ever seen. And I'll be humble to say that I took this home and I couldn't figure it out. I thought she was wrong because I couldn't figure out how this related to the chemistry that we knew. And this kind of goes back to how do we figure out or gain confidence in our structure? It's not simply 2D NMR. There's lots of structures that 2D NMR can all support. We need something else. And so what we think about it as biosynthetic plausibility. How are these molecules being made? These new compounds are clearly related to this one here, ichnosybin A, and we can see how the bicyclic, which we call the andronathines, are related. That blue bond right here is just, it didn't get formed in andronathines. That's one bond. That makes sense. Everything else is there. And so what I would say to students is always try to take a different perspective. I kept looking at the andronathanols like this, but eventually I flattened the 6-member ring, contorted the 5-membered ring, and realized that this is not that different of a structure from our previous compounds. In fact, it's just this red bond right here. That may seem trivial in some ways, but if you're a synthetic chemist, what that red bond is that you're going from this carbon here on the olefin to a methyl group, the least inactive functionality. But since it is one bond, enzymes can do crazy things. And I was like, okay, this makes sense, and we're getting confident in these structures. We've never got any crystal structures. We've never been able to crystallize these compounds. We were able to solve the absolute steric configuration of these molecules. Well, the absolute steric configuration of this one, absolute on the side chain, relative on the ring system. It is very hard to infer stereochemistry from a rigid ring to a side chain, and that flexibility made that not, we were not able to do that. But are these compounds active? I was super excited because you have more complex structures. They got to be more active. And to remind you, ischnosybin A was very active. We did, in the next round of to the psychoactive drug screening program, send a hydrate. We knew it was going to hydrolyze. Is it active? Unfortunately, no, it's not active. So we have to do something with that alcohol. The andronathines and andronathanols were pretty much dead. The andronathanols were completely dead, up to 5 micromolar. The andronathines were somewhat active, 838 nanomolar. I think they're a little bit more active than that. There is a stability issue. These compounds degrade, something with the olefins reacting with the esters. We have never figured out what's going on there, and it took about 8 months for the psychoactive drug screening program to screen our compounds. We also tested a couple of our other compounds that I'm not going to talk about today. These are the gozodesmins, and what I think was really interesting about these is that we actually inverted selectivity. We now have selectivity for sigma 2 over sigma 1. Why that's important is actually sigma 2 is believed to be a better pharmacological target. It's involved in calcium signaling, and there's actually a lot more functional assays with that. And so if you ask any person in pharma, they're more excited about a sigma 2 hit over a sigma 1. And the fact that we have in millipedes hitting both of the sigmas with selectivity, I think gives us a good resource to probe the differences between sigma 1 and sigma 2. Because the andronathanols were completely inactive, clearly we're not hitting the ecological target. A lot of these kind of compounds hit sodium channels, so we did screen our compounds against sodium channels, completely inactive against sodium channels, and that was rather surprising. But I will note that sigma receptors evolved in humans. They're not in any animal out in the field. They're not in the ants, they're not in cockroaches, and so clearly this is a receptor that we're hitting coincidentally, not its ecological target. And so we still don't know what these ecological targets are. But we started asking, who is really the true predator of millipedes? If you look back on Jerry Meinwald's work, who solved a lot of the structures in Polyzonida, the other order, he suggests that the predators are ants and cockroaches. But if you look at the sizes between the millipedes, they're about 3 centimeters or a little smaller than that. Can ants really be a predator for prey? When we were in the field, my group, we noticed that ants and millipedes reside happily under logs together. They're not fighting each other. And if you look at the literature even closer of John Daly's work, you'll notice that poison dart frogs have actually been shown to have some of the alkaloids from millipedes in their skin. And this is work done at Costa Rica. And I could imagine a frog being a predator of a millipede. That size relationship is a little bit better there, where the frogs could eat the millipedes. And in the frog system, it's actually shown to eat this particular millipede here. It's a Polyzonida millipede. And the millipede alkaloid gets sequestered in the skin of the frog. It's not our compounds, but it's the pyrrolidinium compounds. And so we don't have these kind of frogs in Virginia or in California where we collect our millipedes. And so we started thinking about what other predator could this be? And then this video is from Art and Hatch. We are starting to think about salamanders. And so this salamander here is a desmog. And you can see as it attempts to eat the millipede, it clearly has a reaction to it. It's not going to eat this millipede. I should note that the desmogs here are not in the same location as the millipedes. The millipedes like to live in the forest. The desmogs like to live in the creek beds. They're in the same general area. If we do these experiments with the salamanders we find under the same log as the millipedes or near the millipedes, those salamanders don't even try to eat the millipedes. All the times we've exposed them together, they never even attempt to eat them, but they eat the mealworms. And so we think what's happened is that the salamanders that live with our millipedes in the same environment know not to eat these organisms. But if you take one that lives maybe 10 feet away, it doesn't know that, and so it attempts to eat it and clearly does not like it. So cool, these compounds are defensive. We know they bind and engage sigma-1. But more recently, we started wondering, these millipedes have special characteristics. They form multi-generational groups. They form aggregations. Most times, these aggregations are species-specific, even if two species live in the same area. And so we were starting to wonder, could these compounds also be helping identify the millipede self? Could they be identifying, being markers to say, this is my kin or this is not my kin? And so we think that these molecules, we hypothesize that they're dual functioning as both defensive and pheromone. And so to do this, we tried to develop a pheromone assay, and this is all work by Arden, who's here, and her paper just made it out in Journal of Natural Products 2 weeks ago. And so what she did was she took Brachycybie producta, and this millipede here can move around freely. It can go anywhere in this system. And then this millipede here is trapped. It cannot go anywhere. And so it's stuck there. And so what happens is that we're asking Brachycybie producta, who makes gozodesmin, do you want to go towards something that makes gozodesmin, or do you want to go towards an empty container? We also did this experiment where we took a different millipede. We took one from the Polyzonida, so this is a very different millipede, where you have Brachycybie producta making one thing, and this makes polyzonamine. And we asked producta, do you want to go towards something that makes something else, or go towards an empty vial? And so these were done with 30 replicates. The millipedes are nocturnal, so we recorded the video. We recorded the movements with a GoPro camera, and let them go for about 90 minutes and then watch the video. If you just use solvent in both sides, there is no choice preference. Great. There is no preference for where we put this Y tube in the box. If you then say to producta, do you want to go towards yourself, it actually goes towards itself 70% of the time when you use 30 replicates. And this is significant, and so we believe this is being attractant. If we do a different species, Brachycybie rosea, and unfortunately there's no significant difference between which direction it goes. But Brachycybie rosea actually makes significantly more scaffolds than Brachycybie producta, which only makes one thing. And it's also hard to get a millipede into a small little tube, and so we think we're actually activating an alarm pheromone on the millipede. If we do the different species, so this is now going against the Polyzonida that makes polyzonamine, it actually goes away from the other chemistry 70% of the time, rather than going towards it. So it seems to be repelled by having a different chemistry there. And then what was most interesting is that we can replicate these phenotypes with chemistry alone. You can take the crude extract, and you get it going 70% of the time. You can take hydrogozodesmin, the major compound in that secretion. It also chooses to go towards the chemistry, even when there's not a millipede there. And so we do believe these compounds are acting as pheromones, and that might be also why they're able to form these aggregations, but it also might be why we see so much chemical diversity, that it needs to diversify if there's going to be a new species, because it has to be able to identify itself. And so over the past 6 years, we've worked on lots of projects, and you'll notice I think Paul Merrick is on all of these papers with us, and so he's been a great collaborator on that. But we started wondering, how are these molecules made? So if you look at the chemical diversity, it starts to become clear that these things are coming from a common precursor. We think this precursor is geranyl pyrrolidine, shown here. We're not really too worried that there's some 6-membered rings in these systems. That's just, this is coming from ornithine. If it's 6-membered ring is coming from lysine, it's a very simple switch in alkaloid biosynthesis. But when we look at these molecules, there are 2 different ways that these molecules are being folded up. So it makes us think that there are 2 different cyclases. If you look at where this methyl is going, right here, we predict that the methyl and many of these, even andronaphthines, are going opposing the pyrrolidine ring. They're on the opposite side of the pyrrolidine ring. Whereas for the gozodesmins, that methyl is positioned right next to the pyrrolidine ring. And this is just a difference in how these molecules are being cyclized. We were able to take this information and then start to predict what this biosynthetic pathway might look like. And so this is our proposed biosynthetic pathway. We think it comes from geranyl so a monoterpene. And then it's got to do an isomerization where this olefin moves over to the different position there. And then when you get there, then it's a cyclization reaction. And this is well known in terpene cyclization, that you can do a cascading carbocation rearrangement. And the evidence that we have for this biosynthesis is that we have most of the intermediates isolated and structures elucidated. So if you quench the carbocation here, you get deoxypozonamine. You can also protonate, and you get this compound here. We see that by mass spec, but we haven't gone to try to isolate it. And then all you need is an oxidase to generate pozonamine, to generate the ischnosybins. And so it's just tailoring enzymes from there. The andrognathes, the bicyclics, we think that this carbocation here is no longer stabilized. And so it gets quenched at that position, and then it's forming this carbon backbone. And then again, just oxidation and ligation is all that is needed to tailor. We did have the tetracyclic. So this one caused a little bit more problems. To go from here to this tetracyclic, you would have to somehow activate this methyl group. That is not trivial. And so I asked Arden to go back to our millipede collection, and I go look at something really polar. Most studies in the millipedes have been using GCMS, and so you miss the polar metabolites. We use LCMS, but we've never really looked at the polar front portion of our chromatogram. And so when Arden went back and looked at several of our millipede extracts for that, she actually found several compounds that help us figure out this biosynthetic pathway. We now have compounds that we predicted would exist, but we now have functionalization of a carboxylic acid on this gem dimethyl of the terpene. There is oxidation of that inactive center, and now we can propose that this is likely the intermediate we were missing. Maybe not the carboxylic acid, maybe it's the aldehyde, and what we're isolating is the carboxylic acid, but you have an olefin, and now you have an activated center. In some form, it's now set up to make that final cyclization, likely using an enzyme for a cyclase. We do not have any more evidence for this except for the compounds that are in boxes that we have isolated or characterized in some way. But we want to figure this out. And so we're looking for these key genes here, that isomerase, that cyclase, and some of the oxidases. I was a little humbled because I was like, if we can find the oxidases, we can easily synthesize our compounds in the laboratory using chemoenzymatic synthesis. But apparently the oxidases are some of the hardest things to find because organisms have so many oxidases. And so what we chose to do is we chose to use this millipede Brachycybe producta. The reason we use producta is because it lives in the laboratory very well. We've had a culture of it going for about a year, and it's a well-behaving organism. It also only produces 2 compounds, which is our most simple millipede. And so what Arden did is she dissected off the heads. The heads are believed to have no glands, no chemical defense glands. Therefore, they should not have the capability of synthesizing chemical defenses. We took the bodies, and we also took the peronoda. The bodies have everything, the peronoda including, and the peronoda are where the chemical secretion glands are. These millipedes are about 2 centimeters long, so this is not trivial to get these, to dissect these organisms. And what she did was she was able to get 5 samples of pretty much each of them, and we went and did RNA-seq. And we're looking to see how are the genes regulated in each of the samples. And I should note that we were very fortunate that another person in California published a Brachycybe producta genome that was high-quality genome sequenced by PacBio, and it became publicly available recently. And so we've been able to use that information to scaffold our RNA data off of it and to annotate that genome. The genome is 300 megabase pairs, to give you an idea of what we're kind of dealing with here. And so she used a couple different tools to analyze this, but one of them was this new tool called SynBGC. It's a tool that Eric Schmidt developed to find animal biosynthetic gene clusters, which that term is still under some debate of what it's going to be. But what she found was there's actually this one contig that was overexpressed in many of our samples. So the only one that was really significantly overexpressed was this cyclase. But if you look at that cyclase and what is nearby, we actually have another cyclase, and we have a ligase, a oxidation, and we have some isomerases. We have the genes that we need. We may be novel, or we've never done this work before, so we're kind of going in blind. We don't know for sure that this is the pathway. But when you look at all of these genes together and their expression levels, summing these genes together, they're clearly less expressed in the heads versus the bodies in the paranota, which is exactly what we were hoping for. And so Arden has currently purchased all of these genes on plasmids. We are working to express them. I think she's expressed 3 of them, and we need to purify them when we get back. And we're going to test to see, are these the cyclases, and is geranyl the actual substrate for this? And so that's where we're at right now. So I'll just summarize. We've been working on millipedes. We found lots of compounds. They engage different neuroreceptors, and we're excited to be leveraging them towards the treatment of probably pain. We believe the compounds are acting with both defensive properties, probably against salamanders or other amphibians. And we now know that they're also acting as pheromones, and they're dual functioning. There's lots of future work to be done. Millipedes, there's about 7,000 described millipedes, maybe a little bit more than that, but they're under described. Paul told us yesterday that he's done about 150 descriptions of new species, which is phenomenal, but there's probably still 50,000 or more out there. And all of them from the Colanatha group are likely to make alkaloids. Can we find the biosynthetic gene cluster? We're well on our way. We're trying to do that. We have great collaborators for that. If we can find the biosynthetic gene cluster, can we have a microbe make these compounds for us? Because our problem right now is the source of the material. And we really need to get in vivo activity. We can get a stable compound, but do we have in vivo efficacy in an animal study? And so that's what we're trying to get next. We actually just got a grant to do a total synthesis. My group's not a total synthetic group, so we're collaborating with someone at UC Davis, Zhang Wang. And so with that, I'll thank everyone in the group that's done it. I've been very fortunate to have great grad students. We're at our smallest we've been in a long time with 5 grad students and a postdoc. Great collaborators. Many on this project are listed here. And we've been very fortunate about funding. This project is funded with the groups in Mexico, and we're also funded on the discovery of other millipedes in the US as well. With that, I'll take any questions.
Emily. We'll talk.
Nice talking. I really enjoyed it. I have a question. Why do you think, regarding the pyrolysidine nucleus, why you didn't consider lysine as a precursor?
Lysine?
Lysine, yeah, because
lysine.
You have quinolysidines and you have.
Yeah.
Okay.
So lysine is a precursor to the quinolysine.
Yeah, the one that has the 2,6 member.
Yeah. Okay. Yeah. Lysine is the precursor for that. Because more of ours have the 5-member, the inositidine. I've only considered, I only talk about them that way. But yes, lysine is the other precursor, we believe.
And it's very interesting that you have those compounds that are toxic in plants also.
Yeah.
You know, in the Asteraceae family, they are the #1 toxic compound. Of course, they have a different core, but it's very interesting, that you have that convergence, I will say.
There's a lot of convergent with plants, and I think arthropods have probably the most convergence with plants. Now that we're starting to learn more about where some of the frog compounds have been isolated from, and I don't know that connection. If we can find the gene, it would be really exciting to see how similar the gene is to the plants, because they've already figured out those genes in the plants. And so I guess, Arden, actually, you could probably take one of the clisylidine cyclases from plants and see how similar are the ones we're looking at, and did they come from plants?
Yeah, but very interesting, very, very interesting.
We were really surprised by the sodium channel assays. I didn't want to test against sodium channels, because if you hit sodium channels, your drug discovery platform is dead. You can't hit sodium channels and sigma at the same time. And pumalotoxin potently hits sigma, the sodium channels, and
But I think it's a good idea to test all these channels, because insects, that's the target. To kill all the different type of pests. Let's say that way. Invertebrates. I mean, so that's very interesting. That you take. Well, congratulations. No wonder you get the prize from the journal. So thank you.
Yeah.
Thank you for a very inspiring
Thank you.
lecture. Okay.
¿Preguntas?
So, thanks. So basically, Millipede doesn't have predators up to date, or not discovered yet?
I would say we don't know what they are. I'm sure they have something. That's why they would defend themselves. My bet is that they're amphibians. And that's the only reason I say that is because when we turn Colonatha, the ones that live under logs, I don't think their predators are birds. But other millipedes' predators are probably flying because they go on the forest floor. But for ours that are underside logs, we've turned over thousands of logs now. We rarely see a salamander and a millipede together, but we'll see them commonly all over. And so that told us maybe it was salamanders. It's really hard to get permission to work on vertebrates. So to get that video, it took us 9 months of permissions to get vertebrate work. So most people have worked on inverts for that reason. And I think the literature is a little too biased that they're inverts. And I think primarily because it's hard to do invertebrate work.
Thank you.
¿Alguna otra pregunta?
Hi. Thank you for your lecture. I have a question. Did you investigate only for the binding with sigma, serotonin, and dopamine? But I saw the video in you and the animal is like when the bite the worm.
Yep.
Is spicy because did you investigate other type of the receptors like TRP?
Yep. So we tested against 65 receptors. The only ones I showed were the ones that binded 50% or more. And so they were inactive. I will say we do need to go back and do a couple functional assays because there's been some reports that some of the older compounds they don't bind the active site, so they're allosteric binders, and you can only detect that in a functional assay. So we want to go back and do a functional assay of the serotonin because there's a chance that has some activity there. But to do that, you have to tell the psychoactive drug screening program that you want a functional assay, and so we're working on getting more material to send it back down there. They typically like 10 milligrams of a compound. That is very high. I convinced them to take 2 milligrams, but they're not willing to do that for my next round. And so it's just a debate with them of trying to get them to do assays with 2 milligrams. Yeah.
Thank you.
Yeah, you're welcome.
¿Una otra?
The defensive secretion comes out as a liquid, and you said that you put it on the chemical analysis instruments. Can you talk about other parts of the secretion?
Yeah.
Possibly, you said there's possibly monoterpenes and maybe like some sort of other like sticky secrete solution in there or something like that?
Yeah. So we don't know what the liquid is. You're perfectly right. Like our compounds are solids, so they're in some kind of liquid. There is pinene in there, so it could be a pinene. We don't see the pinene on LCMS, but we always see it on GCMS. So is pinene the solvent? Possibly. To see other, like, in some of the other millipedes, it's been reported that it's like, someone reported an ethanol recently is in there. Those analyses are really tricky because all solvents have impurities and their controls also had ethanol in it. And so I don't know if that's strong evidence that there's there, but it could be. These are all got short fatty acids on there. So butyric acid, acetate, those are all liquids too. So it could be the fatty acids are also solubilizing them with the pinenes, but it's 100% not our compounds. And we are missing an element there with what is the liquid component. Yeah. But I can't smell. Everyone says it smells a lot when they turn the logs over, so that maybe there's a lot more pinene in there than we imagine. We could potentially poke a single millipede. We can't inject that on the GC directly. We would have to poke a lot of millipedes to do that. But we could try to poke a lot of millipedes to do that, to get their raw secretions. But we've always put their raw secretion in organic solvents. And at that point in time, I think you lose any ability because those organic solvents are contaminated. Or you got to buy a super high quality grade organic solvent to know that there's nothing else in there. But it would be worth doing because I've never actually thought about that nor gotten that question.
No one.
Hi. About how millipedes identify itself. Have you ever tried to do the experiment without the worm, only with the compound, to see if you have the same results? Or I don't know if it's possible.
No, so just using the crude extract and having the millipede go towards the crude extract or come?
Yeah.
Yeah, we did that. And it actually had the same phenotype.
The same?
We did not do the dissimilar chemistry. So we did not try putting the dissimilar chemistry there and the other side and see if it goes away from the chemistry. The reason for that is we just didn't have enough polyzonida millipedes to isolate that other compound. We thought the reviewers were going to ask us for that. They didn't. So we didn't try to do it.
Thank you.
Okay, la última pregunta del día, por favor.
We don't have another. Emily, I forgot. I'm sorry. It caught my attention the worm I call like
That's fine.
that produces cyanidric acid. Have you tried or other workers the chemistry to see if what they have are glucosinolates? Do you know?
I do not know. Those millipedes have been studied a whole lot better than our colonatha, so I assume that there's probably an answer out there. They just have been. There's a lot more of them. They're easier to find, and people have been studying them for 50 or 60 years.
Yeah, it caught my attention. And finally, why they like the fungi?
Yeah, so that's probably a better question for Paul. But they have really primitive mouths, so they can only eat soft foods, and so we believe that they like suck up their food by possibly excreting some enzymes to break down the fungus and then suck it up.
Okay.
But they don't have mouths that they could chew.
Thank you very much.
Yeah.
Well, Emily, thank you very much again for this wonderful talk, and gracias a todos por estar aquí.
Thanks.
¿Quiere otra pregunta? Bueno, después me pueden buscar y le podemos mandar un correo si tienen interés en ir a Virginia Tech o hacer una colaboración, pues nos podemos poner en contacto. Muchas gracias a todos.
Yeah. I was just telling myself to slow down all the time. I go really fast sometimes.
It was perfect.